website of our World Mitochondria Society. Now, it's my pleasure to introduce our speaker. Professor Howy Jacobs is a distinguished molecular biologist who is specialized in mitochondrial research. Since 1996, he has served as a professor of molecular biology at Tampere University in Finland, and his academic journey includes studies at the University of Cambridge, the University of Glasgow, and he was also a postdoc at the California Institute of Technology. Professor Jacobs' research focuses on mitochondrial DNA replication, mitochondrial diseases, and cellular energy metabolism. And more recently, if I may say so, he has focused his investigation on the elevated temperatures inside mitochondria and the implication of these elevated temperatures on cellular function. And that's exactly the topic he is going to talk about today, about hot mitochondria. Throughout his career, Professor Jacobs has received several accolades, including the European Union's Discard Prize in 2004, and he has been named Professor of the Year by the Finnish Professors Union in 2009. And he also has contributed to the scientific community as the director of the Institute of Biotechnology at the University of Helsinki and as the chief editor of the EMBO reports. In addition to his research and teaching, Professor Jacobs is active in science communication, as it shows today, participating in TV documentaries, public lectures, and writing blogs on scientific topics. I'm extremely excited to have you as our guest speaker today, Professor Jacobs, and I'm looking forward to your talk, and the floor is all yours.
Okay, thank you very much, and thank you to the audience for attending, and very much.
I forgot something. I'm sorry. It's our first webinar. Okay, I forgot something to the audience. Please, any questions you might have, write it in the chat box. And if we have time at the end of the webinar, I will select a few questions and ask Professor Jacobs. And if we get more questions, please email your questions to us, to the World Mitochondria Society, and we will forward these questions to Professor Jacobs, and he has promised us before the meeting that he will try to answer them in due time. I apologize for having interrupted you, Professor Jacobs. Now I'm all set. And
no problem.
Thank you.
Okay, thank you very much. And thank you very much to the World Mitochondria Society for the invitation to talk to you this afternoon. I'm going to try to share my screen. Let's see. Oh, let's have a look. Okay. Okay. Before I start my talk, I'd like to actually apologize for a slight mistake on my part, and which was to schedule this webinar coincidentally with the Gordon conference that's taking place at the moment in California. So if anybody is tuning in from Ventura, it means that you are skiving from the Gordon conference. So please, at the end of my talk, rush away and rejoin the Gordon conference in Ventura. Okay. As I've said, the webinar is organized by the World Mitochondria Society, and so I think it's appropriate for me to start by just making a short plug for the Society and its annual meeting in Berlin, which is taking place this year between the 22nd and 24th of October, entitled Targeting Mitochondria. And as those of you who have been associated with the Society previously will probably know, the aim of the meeting is to showcase latest findings on mitochondrial function and dysfunction, explore strategies for repairing mitochondrial dysfunction in disease conditions, and to discuss ways to target therapeutic agents to mitochondria. And further information can be obtained from the Society's website. So today I'm going to skirt over some published data from my lab and our close collaborators, addressing the question of just how hot are mitochondria. And so the first half of my talk will be published data. The second half, and the published data is really those three references that I put at the bottom. I think that's sufficient for you to be able to dig them out of PubMed. The second half of my talk is going to be addressing the question, what are the implications of elevated mitochondrial temperature for biology and medicine? And I freely admit that most of what I'll have to say will be wild speculation. Some of it is in press and about to be published in a Tibbs article that I've co-authored with Pierre Rustin in Paris and a couple of other collaborators, which we've called the elephant in the lab, the elephant being the fact that mitochondrial heat production has been largely ignored by most people in the field, except where they have been addressing specific questions of thermogenesis. Whereas what I'm going to talk about today hopefully will persuade you at least of the possibility that mitochondrial thermogenesis is important very broadly in biology. So, before we began these studies in collaboration with Pierre Rustin about 7 years ago now, I think most people, including ourselves, assumed that because cells in culture or in our bodies are maintained at about 37 degrees Celsius, the mitochondria inside the cell would also be at 37 degrees Celsius. However, that ignores the fact that the major function of mitochondria is to unlock the chemical energy that is present in substrate molecules, which are then oxidized in an exothermic reaction or series of reactions to carbon dioxide and water, with some of the energy, but only some of the energy conserved as ATP, because the overall reaction efficiency here is only the order of about 50%, according to most estimates. So about half of the energy that we begin with sugars and fats, is actually being released in this process as heat. And this shouldn't really be very surprising if you just consider the analogy of your domestic heating system. Outside, it may be, say, 12 degrees Celsius. In Finland, it might even be more commonly minus 12 degrees Celsius. But the room is being maintained at a constant, comfortable temperature of 22 because the house in which you're living is well insulated and there's an internal source of heat from the central heating system. But if you touch the radiator, it's not at 22 degrees, it's at 52 because it's radiating heat to the apartment in which it's located. And in addition, because of the heat capacity and the insulation properties of the radiator itself, the water that's circulating inside the radiator is hotter still, more like 62 degrees. And so this analogy, I think, should lead us to feel that it's not a great surprise that mitochondria will turn out to be warmer than the cellular environment in which they are present. I think the surprise for us and for many people out there in the community has been that that temperature differential is much greater than we anticipated. So I'm going to just describe the two major methods that we've used to extrapolate the internal temperature of mitochondria. They're both based upon fluorescence, but the basis of that fluorescence is different. In the one case, we load up cells with a mitochondrially targeted temperature-sensitive fluorescent dye. And as illustrated here, and is a little bit counterintuitive for some people, a strong signal from the dye that we've used, MTY, mitothermo yellow, is produced when the dye is cold. And as it heats up, the fluorescence decreases. So pale mitochondria are hot, bright mitochondria are cold. And we track the fluorescence changes in a cuvette by flow cytometry or by microscopy. And we can measure changes in fluorescence according to oxygen supply or the presence of oxphos substrates and inhibitors. And here's just an example of the kind of readout that we get from such an experiment, where we have stabilized cells. I hope you can see my cursor arrow here that I'm moving around on the screen. We oxygenate the cells, which are constantly stirred, and we make sure that the fluorescence that we can observe has reached a steady state. And then we add, in this case, rotenone, a drug that inhibits complex 1 of the respiratory chain. And then with respiration dependent on complex 1 being inhibited, some of the heat production is removed, and mitochondria will reach a new steady state temperature, which we can then extrapolate by means of a calibration step at the end of the experiment. So there, over a few minutes, the mitochondria re-equilibrate to a new temperature measured by a new steady state fluorescence, and the drop in temperature is indicated by the increase in fluorescence. And we calibrate in each individual experiment by doing rapid temperature shifts up and down, and then we can use that calibration to in repeat experiments which are not dependent on the exact amount of label that's been taken up by the mitochondria. And we get a fairly linear response in the calibration experiments that we've done when we use different inhibitors, at least over short periods of time. The new temperatures that are reached in the calibration steps the new fluorescence levels that are reached in the calibration steps are constant, and that applies using different inhibitors, different cell lines, and so on. And we've done some control experiments in response to questions that have been raised by various reviewers, and we're fairly confident that the amount of the fluorescent signals are independent of pH, calcium, membrane potential, peroxide, and other such parameters. When we look at the individual cell, we see, in comparison, say, with the staining of cells by just a simple potential-dependent mitochondrial dye like mitotracker deep red, shown on the right-hand side of the slide, we see much more heterogeneity using MTY. So if you look at this image, you can see that there are some cells here that are relatively faint, whereas others have predominantly bright staining mitochondria, which we would assume are, for some reason, cooler, or the mitochondria in that cell are cooler than this one. And then in some cells, it's clear that there's heterogeneity within the cell as well, because if you look here, for example, the mitochondria in the periphery are relatively faint, but those around the cell nucleus are brighter. So we would infer from that there are cool mitochondria in the perinuclear region, but hotter mitochondria in the periphery. The numbers that we get from these kinds of experiments were much larger than we were expecting. And so we have gone on to validate that by using a second method. The first was developed by our colleague Young-Tae Chang in POSTECH in Korea. This second method, which is based on the ratio of the peak fluorescence of two different fluorescent proteins that are co-expressed from the same construct in the cells. This was developed by Takaharu Nagai's group in Osaka in Japan. And the first generation probes that we've used that do this comprised one fluorescent protein whose peak fluorescence is highly temperature dependent, and another co-expressed with it, which is not highly temperature dependent. And by taking the ratio of these two, regardless of the absolute level of expression of this construct, we can extrapolate the temperature, and it follows, again, a fairly linear relationship. And we've confirmed that. So that was confirmed by our colleagues in Japan on the isolated proteins. We've confirmed it in cells, and we get very similar figures using the two different methods. Interestingly, using the ratiometric fluorescent protein method, we always get slightly lower temperature differentials when we inhibit respiration or OXPHOS. It's not always statistically significant, but we think that this is due most likely to the fact that that MTY, the dye, is retained like other rhodamine family dyes from which it's derived at the interface of the mitochondrial inner membrane or the crystal membrane, whereas the mito-GTEM ratiometric fluorescent protein reporters are distributed in the matrix. And the inference from that is that if the inner membrane or the crystal membrane is the site of heat production, then it's logical that the surrounding mitochondrial matrix will be very slightly cooler. Although, as I said, our actual measurements do not show a significant difference always. Proceeding from that, our colleagues both in Korea and Japan have gone on to make a more refined set of tools. So, for example, in addition to MTY, the group in Korea has now developed thermogreens, a set of dyes which are targeted to different compartments in the cell, so that we can look at the question of how heat is distributed and how the temperature of different organelles and subcompartments of organelles varies when we treat cells in specific ways. The original GTEM has now been developed further. The original ratiometric fluorescent probes had a slight methodological problem in that they had to be illuminated with ultraviolet light, and that meant they couldn't be, obviously UV illumination for any length of time will damage cells and macromolecules, so it can't be used long term to do in vivo experiments. But that's now been superseded by a new family of fluorescent proteins which can be illuminated at wavelengths within the visible part of the spectrum, and again the ratio of the two can be used to extrapolate temperature. The temperatures that we have inferred from these experiments are reflected in the meltome of proteins in different cellular compartments, as determined by others and also by ourselves. In this case, our collaborator in Harvard, who looked at the melting temperature of proteins in different submitochondrial and subcellular compartments. The OXPHOS complexes, where the heat is being generated, have a melting temperature in the upper 50s or 60s, whereas other compartments, as you move out from there, have proteins which have TMs at lower and lower temperatures. So, for example, the cytosol 49, the internal portion of the nucleus 47, and so on. When we inhibit with different drugs that are targeted to different complexes of the respiratory chain, we see a graded response along the OXPHOS system, which we interpret to mean that some of the heat that's generated in the inner mitochondrial membrane is being sequentially produced by the successive steps in oxidative phosphorylation, and that as electrons are being fed in further and further down the trunk portion of the respiratory chain, those electrons then are being used to create more and more heat. And interestingly, inhibiting complex 5 is definitely producing, or it leads to a cooling greater than inhibiting complex 4 or 3. And from that, we infer that at least some of the heat production in the cell is directly dependent upon complex 5, the ATP synthase, or the ATPase, which maybe is functioning in some cells or in some mitochondria in the reverse direction. And the same findings broadly are seen in different cell lines and in a Drosophila cell line that we've studied, as well as mammalian cell lines, although the absolute temperature in Drosophila is about 15 degrees lower because the cells are maintained at a much lower temperature. Interestingly, when we treat cells with various insults, we see that the system is is essentially homeostatic, that when we inhibit cytosolic protein synthesis, for example, as shown on the bottom right, although there's a very slight increase in temperature, the fact that we note that the cell is no longer using so much ATP, so ATP demand has dropped, but temperature is maintained more or less constant. It comes back within an hour or so back to the starting level of about 52 degrees based upon both methods. And when we subject cells to a temperature shock, again, we see a transient effect, but then the cells come back to very close to the mitochondria come back to very close to their starting temperature. We grow cells overnight in different media, requiring dependence on or not independence of mitochondrial oxidative pathways. Again, we don't see any difference in the temperature of the mitochondria. And when we treat treat cells that are expressing the alternative oxidase, which I'll come to in a moment, we see a much lesser degree of cooling from adding inhibitors, implying that the alternative oxidase, which we know provides a bypass for complex 3 and 4 of the respiratory chain, it's only carrying a relatively modest proportion of the electron flow, but nevertheless, it's now able to maintain heat production in mitochondria. We're using, in this case, the alternative oxidase that we have studied for a number of years from the C-squared Siona, although it's been earlier much characterized in much more detail in thermogenic plants. Okay, I'm now going to sort of come on to the second half of my talk, which, as I've already advertised, is very much a speculation or a series of proposals. And I think my main message here is that there are lots of biological phenomena that we need to now address. Obviously, the first one is actually, I've already implied, is to look at different cellular compartments and to try to track the flow of heat originating from mitochondria within the cell and to the outside world. And I'll come back to that towards the end of my talk. So in no really particular order, I'm going to first talk about mitochondrial dynamics, the phenomenon whereby mitochondria are known to fragment into small particulate structures and then to fuse again into long tubes. And although I think there's very good evidence that this is part of a cycle of events that enables the cell to identify and direct into a turnover pathway those components of the mitochondrial network that are dysfunctional, that doesn't explain the whole story. And in fact, both fusion and fragmentation of mitochondria are seen under different circumstances where you might expect that the process would be in one direction only. So, for example, on the left, if cells are treated with an inhibitor of complex 1, metformin has other effects in cells as well. And these, I believe, are U2OS, osteosarcoma cells. Then mitochondria fuse into these long tubular structures. Whereas if cells are treated with an uncoupler, such as CCCP, then they fragment into tiny little blobs. Now, we don't know what, as I said, we don't have a full explanation of what's going on in these cases. But the proposal I'd like to throw out here is that because heat dissipation from mitochondria should be influenced by the surface area to volume ratio, then because the transition between tubes and fragmented particles changes the surface area to volume ratio, it may be related to heat production. And we know in cells, because it's been measured by our collaborators in Japan, that when cells, and we've also done the same experiment ourselves, although it's not been published in Drosophila cells, when we treat cells with an uncoupler, according to the ways in which we've measured mitochondrial temperature, mitochondria heat up and they fragment. And that's consistent with the idea that the mitochondria are overheating and are trying, in a sense, to dissipate that extra heat production beyond the mitochondria by increasing their surface area. On the other hand, when cells are inhibited from respiration from specific respiratory complexes, such as here, complex 1, then they start to go cold, as we've shown in a number of cell lines when we treat with inhibitors of complex 1. And the result is that the mitochondria are getting cold, so they, in a sense, like the penguins on the ice cap in Antarctica, they're getting together and reducing their overall surface area for a given volume. So we think this can be measured, and I think I'd like to suggest that somebody would do these experiments in a systematic way to look at whether surface area to volume ratio is really a determined, or is really being determined by mitochondrial heat production. In the same vein, mitochondria, we know, move around inside cells, especially inside neurons, and that has been traditionally considered to be a process by which mitochondria deliver ATP and metabolites to the cell periphery, and then return to the perinuclear region to take up new substrates or to be replenished with proteins that have become turned over. And so this whole process is again connected with the maintenance of mitochondrial quality. However, it's also possible that this process has evolved to supply heat to the periphery of the cell, so that as mitochondria travel, for example, through an axon to the synapse, they're then able to supply heat to maintain the processes that are taking place in the synapse, the synaptic transmission of neurotransmitters at the constant optimal temperature. Another topic that's caught our attention is that of immunity. So I'm not an immunologist any more than I'm a theoretical physicist, but my understanding of what cytotoxic cells do from the literature, from a large number of studies that have been done, is that there's some intimate association of cytotoxic cells, as well, of course, as phagocytic cells, with pathogens or cells that are infected with pathogens, and the delivery of something into the target cell to ensure its death, the death of the pathogen or the pathogen that it has harbored. And in general, although there's no completely agreed, as far as I understand it, there's no completely agreed understanding of what it is that's transferred. There's some evidence that what is transferred are pro-apoptotic signals, reactive oxygen species, which then can damage the contents of the target cell, and perforins, proteins that puncture holes in the target cell membrane and cause it to lose its cytoplasmic constituents and become unviable. But there's also increasing evidence that mitochondria are clearly involved in this process. And so one possibility is that the mitochondria are themselves delivering something through the synapse that is formed with the target cell. And one of the things that mitochondria might be supplying here is heat. The heat could be supplied either in some chemical form as a result of the mitochondrial process of heat release, and we'll come to that perhaps in the end of my talk, or mitochondria themselves are being transferred through nanotubes or extracellular vesicles, and there's increasing evidence that mitochondria are indeed being delivered or exchanged between cytotoxic cells and virally infected cells in the process of cell killing. Of course, this can also involve not just the transfer of mitochondria, but the transfer of something that leads to the uncoupling of mitochondria in the target cell. In other words, the hypothesis here is simply that the cytotoxic cell is inducing the target cell to overheat and that's part of the process by which the target cell is killed. And I think that's also a neater explanation for the phenomenon of fever than what has been traditionally understood. Most people seem to have believed that fever is a manifestation of a response in the body to generate heat to kill pathogens that are circulating. But the amount of heat that we're talking about in fever is only a few degrees Celsius. And we know that mammalian cells are much more sensitive to that kind of heat, that kind of temperature increase than the pathogens that the immune system is trying to destroy. So fever alone should not eliminate the pathogens and might actually favor them. And that's also reflected in the meltome of typical intracellular bacteria, such as, E. coli is not intracellular, but it's present in the body, being maintained at 37 degrees, but nevertheless, its meltome is much higher. Its TM of its proteins is much higher than 37 degrees, in the same range as mitochondria. So my suggestion here is that the manifestation of fever is simply the leftover heat from this thermogenic cell-killing mechanism. So the target cells are being fried, and the heat that's then released into the circulation is what we perceive as fever. Aerobic bacteria in general have a very similar energy generating system as the one found in mitochondria. That's not surprising because mitochondria arose in the course of evolution from the entry or incorporation of aerobic bacteria into the eukaryotic ancestor. And based on that similarity, I think there's a good chance that since aerobic bacteria and mitochondria have essentially the same heating system, and also both have a double membrane, or in the case of gram-positive bacteria, a very thick bacterial cell wall, they both have insulating layers which enable this heat, so some of the heat, to be retained within the cytoplasm of the bacteria. So my proposal or prediction here is that when people get round to actually measuring the internal temperature of bacteria using the same kind of tools that we've used for mitochondria, they'll get a similar surprise and find that bacteria with their layers of insulation, even if they're living at 37 degrees or at 15 degrees or wherever they are in the environment, are internally much warmer than the environment where they're living. Although this is not directly my idea, it's inspired by a colleague of mine, Corey Dunn, who was in Helsinki for a number of years. But the production of heat by aerobic bacteria and subsequently by mitochondria might provide a neat explanation for the original endosymbiotic event which led to the formation of eukaryotes. So traditionally, people have thought of the marriage of aerobic bacteria and a primitive non-photosynthetic and non-respiratory, in other words, anaerobic archaeon as being due to the fact that the bacteria were delivering ATP in a much more efficient fashion. However, this alternative hypothesis postulates that originally the archaea that gave rise to the eukaryotic host were living in a warm portion of the ocean around hydrothermal vents, so they were thermophiles, whereas the aerobic bacteria that evolved were able to live in a much cooler environment at the cell surface where they had access to more oxygen to do respiration, living off some of the products that were floating around in the ocean that were being produced by the archaea and other organisms, but they were producing heat which enabled them to live efficiently in a much cooler environment. But by the thermophilic archaea getting together with aerobic bacteria, the archaea themselves could float up to the surface and live in a much cooler environment because the heat to which they were used in their original environment was now being supplied externally by aerobic bacteria. And in time, the suggestion is that those heaters that were acquired by the originally thermophilic archaea were internalized, because that then provides a much more efficient delivery of the heat to the eukaryotic ancestral cell. And in support of that, it now appears that based on some work was done last year in another lab, that the extrapolated eukaryotic ancestor, the original archaeon that absorbed or became associated with aerobic bacteria that then turned into mitochondria, was indeed a moderate thermophile, and its proteins are optimized for life at around 55 degrees. Which leads me to another evolutionary issue, a long-standing one, which is the question of why mammals and birds, regardless of their size, their living environment, or other aspects of their metabolism, are all maintaining their body temperature around about 40 degrees Celsius. We know, for example, that human cells can't tolerate temperatures above about 43 degrees for any length of time. And we talked a few moments ago about fever. Whereas some other animals, such as lizards, are quite happy at temperatures fluctuating between 0 and plus 50 degrees. And one suggestion that I think might explain this phenomenon is based on the fact that this event that gave rise to mammals and birds was probably a rather rapid event in evolution when the ancestor of land-living vertebrates crawled out of the ocean. And the idea then is that rapid coevolution of the 20,000 or so protein-coding genes would have been very unlikely in a short period of time. However, just mitochondria increasing their heat output, for example, by becoming less stringently coupled, or having more mitochondria per cell, heat production in that creature could be greatly enhanced in the cooler environment, which we know occurred about 290, 300 million years ago, based on the geological record. And interestingly, geochemists who have been extrapolating the temperature of the ancient ocean during the evolution of our planet have inferred that the temperature of the ocean at the time that the vertebrates came onto land was about 40 degrees Celsius. In other words, what was being provided by the warm sea was in the cooler air where the ancestors of mammals and birds and dinosaurs were living, were now being provided, that heat was now being provided instead from another source, an internal source, and that obviously is likely to have been mitochondria. Other creatures living in the sea didn't adopt this homeothermic mechanism. They didn't need to, because they maintained their body temperature over a long period of evolutionary time. As the oceans cooled very gradually, that was sufficient time for all the proteins encoded by their genome to evolve, to adapt to a lower temperature environment. So sea urchins and fish and so on living in the ocean are quite happily living at about 10 or 12 degrees now instead of 40. Mitochondrial heat production may also be very important in disease. The very first mitochondrial disease that was first reported in the 1950s by Rolf Luft in Sweden was, in fact, a kind of malignant hypothermia, where muscle was overheating, but at the same time being starved of real energy because the mitochondria in that muscle were effectively uncoupled. And somehow that's all been set aside and a little bit forgotten about over the years. And mitochondrial dysfunction in cases of disease has been considered to be due to insufficiency of ATP, to redox imbalance, excess production of toxic oxygen radicals, or imbalances in metabolites or ions, and temperature has been ignored. However, I think we should perhaps go back and look more carefully at the possibility that mitochondrial temperature anomalies underlie some of the features of mitochondrial disease. And there is at least one, I think, credible paper out there looking at the temperature of brains of mitochondrial disease patients, identifying a hypothermic response. In other words, that when mitochondrial OXPHOS is not working efficiently, is not working adequately, then the provision of heat is inadequate and the tissues go cold. In the heart, which is also a target organ in a number of mitochondrial diseases, ATP is required for cardiomyocyte muscle contraction. The ATP is obviously generated principally by mitochondrial substrate oxidation, which, as I've already said, releases a lot of heat. The pumping action of the heart itself then converts that ATP into useful work, but also again releasing some of the energy as heat. And the pumping action of the heart itself contributes to delivering oxygen and nutrients to the tissues, but also in taking heat away from the heart. That actual heat that's being output from the heart needs to be removed, and the pumping action of the heart is doing that. But of course, if either of these processes is disturbed, if mitochondrial substrate oxidation becomes less efficient, producing more heat for a given amount of ATP, or if the pumping action by muscle contraction of the heart is no longer doing enough work but is releasing too much heat, then a vicious cycle is going to result. Too much heat is produced, extra pumping is needed, and eventually even more heat will be produced, leading to a catastrophic overheating of the pump. And that might, in fact, be something that underlies heart failure. I'm reaching the end of my talk, so I'm going to go quite quickly here. We know already some examples in the literature, clearly where specific cell types or organs of both mammals and thermogenic plants repurpose mitochondrial respiration to generate less ATP or no ATP and much more heat. The brown fat of mammals, the reproductive organs of aroid lilies, which volatilize insect attractants by the production of mitochondrial heat. And we've suggested that a similar process might be taking place in the Antarctic sea urchin, where protein synthesis is very active following fertilization at similar rates to those seen in temperate species, implying that the whole machinery of protein synthesis is somehow behaving as if it is a much higher temperature. And we would like to look to see if that actually is borne out, if the internal temperature of the zygote and then the early embryo, and in particular the mitochondria, is being maintained at a much higher temperature than the -1 that the organism is living at, that the animal is living at. Of course, when these thermogenic systems operate, the cells in which they are located, the cells that are producing the heat, are not destined just to become hot. They have to deliver the heat to the rest of the organism. And therefore, they need to protect themselves against overheating, because if they're delivering heat to the outside, either that delivery's got to be very efficient, or they will risk becoming far too hot internally. We know one of the methods by which they can do that is by producing heat shock proteins, but their ability to resist high temperatures is not unlimited. So an alternative proposal is, and we have no really strong evidence that either of these exists, that there must be some efficient mechanism for transducing heat from the source, from mitochondria, to the outside without the cell itself getting hot. For example, by a series of conformational changes of the cytoskeleton, which then enable the heat to be pushed out of the cell, or perhaps more plausibly, by the generation of a heat store molecule, a thermochemical, which is produced by an endothermic reaction, gets out of the cell, and then undergoes a reverse exothermic reaction, so that the thermochemical itself is carrying the heat from the source to the place where it needs to be delivered. And that's also the principle that applies in thermosolar power stations. I'm going to end by suggesting also that heat release may also be involved in the process of apoptosis, and this may resemble what's going on in cytotoxic events in the immune system. We know that mitochondria are a source of one of the major signals in the life-death decisions of cells. And we know that even in in organisms where the activation of the caspases doesn't seem to be crucial for cell death, including plants, that there is nevertheless a large release of cytochrome C from mitochondria. And we know also that cytochrome C is released from mitochondria in an oxidized form, but in the cell cytosol becomes reduced. And this itself should be an exothermic reaction. So one possibility is that the oxidized cytochrome C that floods out of mitochondria when apoptotic signals puncture holes in the outer mitochondrial membrane is then when it gets out of the mitochondria into the cell cytosol, causes a rapid increase in temperature, which then in effect fries the cell. I'm going to finish just by alluding to a couple of objections that have been raised to our extrapolated temperatures. As I said, I'm not a theoretical physicist, so I don't have a complete explanation or a complete understanding of the physics behind this particular proposal. But some people have suggested that the temperature difference that we have implied between hot mitochondria and cool cytosol, although strictly speaking, we haven't actually measured the temperature of the cytosol, that sharp boundary cannot exist in nature. But I think that's probably based anyway on a misunderstanding, because there needn't be sharp boundaries. There are a number of compartments across which heat can flow, including the crystal membrane, the intermembrane space, the outer mitochondrial membrane, the perimitochondrial cytosol. So the extrapolated limits of heat, of temperature differences across such boundaries, which is estimated by physicists at about 5 degrees, will probably be brought about collectively by these different barriers to account for the overall 12 to 15 degree or more degree difference between the mitochondrial membrane and the outside of the mitochondria. But a work that was published a couple of years ago by an Italian group demonstrates that Fourier's law, which predicts the distances over which temperature gradients can be maintained, does not actually apply at nanoscale, because we need to factor in somehow the heating effect of the OXPHOS units on each other. Another work that's been published recently from Tonio Enriquez's group in Madrid showed that when isolated mitochondria are heated at more than 43 degrees, the supercomplexes, which are the associations of the different respiratory chain complexes with each other, fall apart. And indeed, some of the so-called supernumerary subunits of the OXPHOS complexes themselves also dissociate. And that has been suggested as evidence that our extrapolation of high mitochondrial temperature might not be correct, because it's assumed that the supercomplex state is the natural state, and that the heating, which produces the dissociation of these supercomplexes, is some kind of in vitro artifact. But of course, it might be the other way around. The individual complexes might be the natural state of hot mitochondria, and they only get together into this perhaps more efficient way of transporting electrons and protons in the cold, because it's cold, and that might be an in vitro artifact because mitochondria are rarely cold. Or I think more plausibly, if that's correct, this could be a stress response to excessive mitochondrial cooling. In other words, it's a homeostatic response when, for whatever reason, the heat production in mitochondria becomes too low to sustain a normal high temperature. Respirosomes, supercomplexes can then form and produce more heat. So that's my suggestion to explain that. And I think we need to do a lot more to study this system to try to understand what's going on, to see who's right and who's wrong. I'll end by acknowledging the people who've done most of the work. The Dai family, developed by Young-Tae Chang, the ratiometric fluorescent probes by Takahara Nagai. All of our work has been done as a collaboration with Pierre Rustin in Paris. And the Meltome work has been done by Dan Davidi in Sterling Churchman's lab in Harvard. Mugen Tessiolu has been my long-standing postdoc doing much of the work here in Tampere, and she is now working as a co-PI on the project in the lab of Tina Salminen, who's also interested in immunity and using the Drosophila model. I'd like to acknowledge my sponsors as well as some other collaborators and those people who I've stolen various images from my talk. And I'll leave you with a summary slide from what I've been saying. So, in other words, mitochondria are about 15 degrees above ambient temperature in diverse cell types based upon the temperature drop measured by these fluorescent probes when we deliver inhibitors. Mitochondrial temperature is homeostatically maintained wherever possible. And then various speculations that mitochondrial heat production may be connected to mitochondrial dynamics, mitochondrial motility, immune cytotoxicity, and apoptosis, and may also account for some evolutionary phenomena as well as some of the features of mitochondrial disease. So I'd like to thank you for listening to me, and I think it's now appropriate to take some questions. Maybe I'll stop sharing in a moment whilst you've taken in what's on the summary slide, and then we can see each other to address the various questions that you may have.
Well, thank you very much, Professor Jacobs, for this highly interesting, if I may say, hot talk. And the facts are surprising. And I think, especially the speculations at the end of your talk, are partially very provocative. And I believe you have triggered a lot of future research avenues, and the time will tell whether your speculations are founded or not. So it was highly interesting. But this was just my statement. You don't have to comment on this statement.
Well, I agree, other than saying I agree with you.
Okay, good. And then we got a few questions, and let's start. Some of them you might have answered already to some extent, but I start with one question. By the way, I mean, also comment, great talk, okay? Now, if mitochondria are hotter than the rest of the cells, what about the mitochondrial proteins, membranes? Are they more heat resistant than the other ones? And this also added something. I mean, you gave a similar talk already in Berlin, I remember, and of course, and you even raised the issue that classic biochemistry, enzymology, took for granted that all enzymes work at 37 degrees centigrade. So that means the whole enzyme kinetic might have to be revisited. But again, the question is, are mitochondrial proteins and membranes more heat resistant than other cellular components?
Well, I think the Meltome studies that I've referred to certainly suggest that mitochondrial proteins, especially those of the OXPHOS system in the cristae, are indeed more thermoresistant than the bulk of cellular proteins. And what you've just said, or the question has implied that if we've been studying mammalian mitochondria or mammalian mitochondrial enzymes at 37 degrees, we may have been subjecting them to totally the wrong temperature and extrapolating wrong inferences from that. One thing that I should also maybe add to the discussion of the results from Enriquez's lab is that respiring mitochondria, when they're heated at high temperature, the actual proteins are even hotter because they are the ones if they're functioning, that they are actually producing more heat, so that when they're subjected to a temperature shock from the outside, they may then become overheated at 60, 65 degrees instead of 52. And as a result, that may then cause them to malfunction. So I'm not going to stick too closely to my hypothesis. It could be wrong, but I think that's an alternative view. And I think work that we've done, or rather that's been done in Paris by Pierre, strongly suggests that the temperature optima of some of the key respiratory enzymes, especially complex 3 and 4, is indeed, and also complex 2, is in the range of 55 degrees, implying that that is the optimal temperature then for OXPHOS.
Thank you very much. We have only 2 or 3 more minutes left, and I pick maybe one question out regarding mitochondrial therapies. I mean, the question is, have we potentially misinterpreted the efficacy of mitochondria-targeted bioactive compounds because we underestimate the temperature of mitochondria? 37 versus 50 is a huge difference, okay? And could this explain maybe also the limited translational success?
Yes, I think that's very plausible. I mean, I've talked a little bit about bacteria, and so if I can just go back to bacteria for a moment, a lot of the drugs that people swallow for various different infectious diseases are antibiotics that target bacteria, and especially to ribosomes of bacteria. And if those ribosomes are functioning at 50, 55 degrees inside bacteria, then efficacy of antibiotic therapies is also actually optimizing action at high temperatures. So, and that may apply also, as you suggested, to drugs and other agents that are targeted to mitochondria.
One more question, if organizers give me the time. I mean, since complex 1 dependent respiration was shown to be sensitive to temperature increase across different species, the question is, why do you think it is important to have such a thermolabile enzyme at the center of heat production?
Well, I think it's questionable whether it is at the center of heat production. It's part of heat production, but it only receives a proportion of the electrons that are eventually transmitted to oxygen in respiration. Electrons are fed in at complex 2 and other dehydrogenases, complex 3, complex 4, and possibly other sources as well. So it's not really at the center. I think we didn't, or rather Pierre didn't succeed in demonstrating in his experiments a high thermoresistance of mammalian complex 1. I think I'm not really an expert in bioenergetic analyses of the individual complexes, so I think I would just fall back upon what I've implied, that the heat production of the individual enzymes may be an important factor in measuring their actual thermal optima. And we need to take that into account, as well as their very specific lipid environment, which may also be crucial.
Okay. Well, we are out of time. And at the end of our webinar, I just would like to thank first Professor Jacobs for his excellent talk. And I would like to thank our team in Paris, who has organized and set up this webinar. And of course, last but not least, I would like to thank all attendees who have participating, almost 140 attendees. And maybe many of them we might see eventually in Berlin in October at our 16th World Conference on Targeted Mitochondria. I would be very happy to see you there. Yeah. Professor Ideas, do you have one more comment?
First of all, I want to thank you very much for this excellent, and I want to thank Professor Jacobs for Fantastic talk, fantastic description. You know, we travel inside the mitochondria and 50 degrees. And thank you again for this fantastic talk. Really fantastic one. Thanks, Volkmar, for this moderation, for the nice question, and for everyone. Thanks for all attendees, and see you next webinar will be about mitochondria transplantation, I think in 2 weeks. Yes, and thank you very much.
Thank you very much again to
Thank
the
you.
audience and to the organizers. Thank you.
Our meeting is now closed. Thank you.
Thank you.